Hey, my name's Christian Stevens and I work in the Haushalter lab. I'm a rising senior here at Harvey Mudd College and a joint biology and chemistry major. In the Haushalter lab, we actually do HIV gene therapy. And so we have a bunch of different members in the lab who work on different parts of the project. My specific part of the project is dealing with what's called a TAR decoy. What we do is we take what HIV wants to do naturally and we mimic it in order to basically trick the proteins that HIV has into binding with our therapy instead of itself like it wants to. And that kind of disables it from being able to transcribe itself and produce more HIV. What we can then do is taking that one really exciting therapy, we can add it to a carrier molecule, which allows us to release different amounts, right? So if you end up having high toxicity, even though it's really effective, you can lower the amount of therapy release in a very easy way. That way in clinical trials, the doctor will have a lot more leeway to work with. And then we can get really tricky with the carrier molecule and add a second therapy at the same time, which allows us to target a whole nother set of HIV's life cycle, which is really, really exciting. So that's kind of the gist of what I personally do in the Haushalter lab. What that involves on a day-to-day basis is producing DNA that codes for all of those therapies, and then taking that downstairs where we actually test it in real human T cells. And so that's the most exciting part, is being able to see if the tests work out or not.